Okay, so we can proceed from where we had left. So welcome again. This is the lecture videos on bacteriology, and please kindly feel free to share, like, and subscribe to our YouTube channel. Okay, so we had stopped at the cell wall, and we're talking about the peptidoglycan layer, and this is a very important layer, especially when you are differentiating into the two major groups of gram-positive and gram-negative. So we will look deeply into this later on when we do gram staining, but for now we just need to know some basic things. The most important one is that for gram-positive bacteria, the peptidoglycan layer is thick. Okay, it's very thick compared to gram-negative, which has a very thin peptidoglycan layer. Okay, so with regards to the membrane, the gram-positive bacteria does not have an outer membrane. Okay, but a gram-negative has an outer membrane, so it has like two membranes. There's the normal plasma membrane, and there's an outer membrane. Okay, so those are the two major issues I would want you to get here. So gram-positive thick peptidoglycan, gram-negative has a thin peptidoglycan layer. Now the rest is basically on constituents, like what exactly does it have. So we have peptidoglycan, the one we talked about, teichoic acid. Now gram-negative has on top of peptidoglycan and the rest, it normally has the lipoprotein, phospholipid. Remember this, they form part of what is going to be an outer membrane. Okay, so this is basically what we're talking about. So if you're keen enough, you can see the size of this peptidoglycan layer. It's thick in a gram-positive, and in a gram-negative bacteria, this is the peptidoglycan layer, very thin comparatively. So and then on top of it, we have this layer of cell membrane. So this is an outer membrane, which has the bilipid layer, and also there's a plasma membrane. So you see we have two membranes. So there's an outer membrane and the plasma membrane. Gram-positive does not have an outer membrane. Okay, just the normal plasma membrane that we have. So this is going to be a very important concept when we'll be looking at gram staining and why that technique works. Okay, and it is basically based on the fact that one has a thick layer of peptidoglycan layer, and then this other one has a thin one, and then it has an outer membrane. So that is basically the major difference between a gram-positive bacteria and a gram-negative, which continues to other aspects, like even pharmacology. Some certain drugs work better in gram-negative bacteria than gram-positive because of these differences. So after the cell wall, we go to the cell membrane. Now the cell membrane is also called the plasma membrane, if you want to call it, or the cytoplasmic membrane. So it is composed of protein and lipid layer. Okay, and then there is also a carbohydrate element, so which forms now the phospholipid layer. Okay, and we will look at it. So it's mostly composed of protein, lipids, and carbohydrates. It's basically what you're talking about. So if you take a look, we have lipid bilayer. So we're calling it bilayer because there's a lipid layer here, lipid layer up here. Okay, then we have the proteins, okay, which normally have which form the transport channels, and then we usually have the carbohydrates now hanging on top of them. Okay, so this is basically what we're calling a cell membrane. So it has a bilipid layer and proteins and carbohydrates. Okay, so the function mostly, as you've seen from that image I've shown you, is it regulates the transport of nutrients and waste products. And this normally works through this transport channel, and also lipophilic constituents can actually go through. Okay, so it's the cell membrane is basically almost it has some level of permeability, so it can allow things to come in and out. So it is not as rigid as the cell wall. Okay, so it also synthesizes things for the cell wall, the components. So whatever you talked about the cell wall, like the different parts of the peptidoglycan layer, the constituents are made from the from the cell membrane. Okay, so the cell membrane helps in the formation of constituents of the cell or the cell wall. Okay, so that is basically what the cell membrane does. Then, in looking at some of the most important cellular elements, we have mesosomes. But just to be clear here is that we have so many types of bacteria, and all these bacteria have, it's not like they're all the same in terms of the constituents that they have. So they might get some differences here and there. But what we're looking at is basically the common cellular elements. So as I said before, mesosomes, they function mostly in cell division, okay, or excretion. If they want to do exocytosis, they want to remove some elements from within the cell outside that formation, that vacuole, and then removes. Okay, and then so if you have your bacteria here, and it forms that vacuole and vacuole. So this one can come and meet up, and then helps in now us having two bacteria that have actually divided. So they help in cell division. Okay. So then we have ribosomes. I think that is obvious for protein synthesis, formation of different proteins that are needed by the bacteria. And then also different proteins that form its genetic material. Then cytoplasm is where we have the nutrients, and then the nucleoid has the genetic material. Okay. So when we go now to the outer part, we are talking about the glycocalyx. And with regards to the glycocalyx, the main elements we look at is the capsule and the slime. So the capsule, as I said earlier, has a firm adherence to the cell wall, basically. Okay, and then it forms this very strong. And but then the capsules usually have the ability to resist phagocytosis. Okay, so capsules have it adds a layer of protection to the bacteria. So in this case, you can see this is the cell wall here, and then the outer part capsule. Then the slime is easily washed off. It's not firmly adherent, so this is basically what you're talking about. This can easily be washed away from the surface, so it does not give a lot of protection. So we also said we have a flagella on the outermost part, and these are flagella. You have different kinds of flagella, so you can have one, now several, you can have on different sides, or you can have all over the bacteria. Okay, so it is a whip-like structure, and it is the organ of locomotion. Okay, and the ones that you're seeing here are external flagella. We can have an internal flagella like in like I think Treponema or the spirochetes. They actually move with internal. They have internal flagellas. Okay, so these are normally for locomotion. Then we have pili. These ones are these hair-like structures that are found on the outer surface, and they're normally composed of proteins. Okay, the pili are made up of proteins. So the structure is most built for adherence on the cell surface. Okay, so when the bacteria is coming into your surface, for example, it is on the mucosal surface or the skin or whatever, so that structure that helps it to settle down and adhere and attach, mostly it's the pili, or what they call the fimbriae. Okay, so that is basically one structure. Okay, so with regards to size of bacteria are comparatively to like viruses, bacteria are bigger than viruses, but they're also smaller than like most fungi or protozoa. Okay, so anyway, the average diameter of bacteria is between 0.5 and 2 micrometer, and then forward shaped um, around 1 to 10, okay, in length, and then diameter is 0.25 to 1. So anyway, the bacterium Pelocysium physiolosoni can be seen in naked eye. You don't need a microscope. So that shows it's a very, very big bacteria. Okay, bacteria that you can see without actually using a microscope is very big. Then we have mycoplasmas, which are the smallest known so far. Okay, but more and more bacteria keep getting discovered, so we might have even a smaller one. Okay, so this is just to show comparative size. Okay, so like these are anatomy is the smallest element known, and then we have like viruses here, see 100 nanometers. Then now we have bacteria, okay, and then other eukaryotic cells. Okay, so in terms of small size, virus is mostly smaller than bacteria. So with regards to size, nothing much, just to know that most of the bacteria we need a microscope to see. And by the way, they are bigger than viruses. That's why bacteria they were the first to be discovered in the microorganism compared to viruses, because the light microscope was the first to be discovered. So they first viewed the bacteria and they thought they had seen all the microorganisms. Now, in the advent of electron microscope, now they were able to see viruses. Okay, yeah. Then, so classification of bacteria, um, we have depends with how you want to classify it. We have different kind of classification, but we mostly focus on the structure, morphology, how they are arranged, staining, and oxygenate. So in terms of their shape, the morphology, arrangement, staining, and bacteria can be divided into all these different types. Okay, but mostly clinically. Okay, because this is basically theoretical. Clinically, the main types of classification of bacteria is based on the morphology and the staining. Okay, so in morphology, the shape we're talking about, we have so many different kinds of shapes, but a cocci is basically a round or an oval shaped bacteria. Okay, so cocci is a round or oval shaped. Bacilli, they are rod shaped or basically cylindrical. Just remember bar. Okay, Coccobacilli is now the almost formation of the two, like amalgamation of the two. So you have something that looks like it's short rods. It's almost like round, but it's also like cylindrical. Then we have spirillum, which is spiral shaped, which is not flexible, and then we have spirochetes. These ones are spiral shaped, but they're flexible. Okay, we have a class of bacteria called spirochetes, by the way, and it's exactly how they are. They have Vibrio, which are comma shaped. Okay, so these are basically what we're talking about: coccus, bacilli, coccobacilli, fusiform, which has two pointed areas, but almost like a bacilli, Vibrio, spirillum, and then spirochetes. Okay. Now the other classification based on arrangement, it's just the same, the different shapes, but now how are they arranged as we move from like, are there so many clustered together? Do they occur as one? Do they occur as two? Do they occur as a chain? Okay, so we have single cells, just a cocci, for example. And then we have diplococci. Okay, this like especially like nice areas. They normally like occurring in form of diplococci. We have tetrads, which occur as four. We have streptococci, which occur as a chain. Okay, like strep means a chain, and then cocci are the round shaped. And we can have staphylococci because they occur as clustered. Okay, staphylococci, they're clustered together. So that's why they're called staphylococci. Okay, so these are very common. Like diplococci are common. Neisseria meningitis or Neisseria gonorrhoeae. Streptococci, you must have heard of Streptococci pneumoniae. They occur as a strep and chain-like. And then we have like Staphylococcus aureus. Those are their formation. Also, we can have the arrangement in a bacilli state like a coccobacilli, diplobacilli, streptobacilli. Okay. Now, with regards to staining, we have so many different types of staining, but we'll talk about just the gram-positive and the gram-negative. So, if you remember what we said about gram-positive bacteria, it has a thick peptidoglycan layer, and with that, it is able to stay with the stain. Okay, the crystal violet stain, which stains purple or blue. Okay, so when you look under a microscope, so you can see these are bacilli. These are bacilli which are blue in color. So these are, if you were to define this, these are gram-positive bacilli. Okay, bacilli. And then we have gram-negative bacteria. These ones have a thin peptidoglycan layer, therefore they normally lose their primary stain. So, and then they stain with a counter stain. I know it sounds weird, but we'll do gram staining and you'll understand the process. So it gets the color of the counter stain, which is saffronin, and it's normally pink or reddish in color. So this is, as you can see, these cocci. You see like the grains all over. So this is like these are cocci and they are red in color. So these are gram-negative cocci. So it depends. If you had a better image, you'll know whether it's a diplococci or staphylococci. Good. So these ones are normally used so much. We have other staining techniques that we'll use like acid-fast bacilli tests. Okay, all those testing are good for diagnostics. So we have examples here of gram-positive. Will not belabor the point here so much. We will look at all this when we do staining, but just examples of. So we have mycobacterium, even though in actual sense mycobacterium don't classify them as gram-positive. And then we have Clostridium, we have Staphylococcus, Streptococcus. Gram-negative, we have so many. We have Salmonella, Bordetella, Chlamydia, E. coli, H. pylori, Klebsiella, Neisseria, all those ones are gram-negative. Okay, so the classification is based on oxygen need. So these ones we can divide mostly into 4 groups. So we can have an obligate, anaerobe, facultative, anaerobe, obligate, or microphilic. So an aerobe basically means that the bacteria can only grow in complete absence of molecular oxygen. Okay, so this is absence. That's why they're called obligate, because they can only grow in absence of molecular oxygen. Okay, so examples are like Clostridium. Like Clostridium tetani, which causes tetanus. Then we have facultative anaerobes. So these are also anaerobes, but facultative means that they can live and grow with or without molecular oxygen. So you actually pay attention that what we are focusing is molecular oxygen, not just oxygen. Okay, because it needs some sort of enzyme to actually break down this oxygen. So when it lacks this component of breaking down oxygen into the one that can be used, then that it becomes an anaerobe. Okay, but when it has these enzymes that can break down the oxygen into a form that can be used, then it is termed as aerobe. Okay, so these are examples of staph species, strep, salmonella. All these are facultative anaerobes. Then aerobes, these ones are bacteria that need oxygen. Okay, because they cannot ferment or respire anaerobically. So these ones require oxygen, okay, for them to survive. A classical example would give, especially for obligatory or obligatory like Mycobacterium tuberculosis. Okay, like that's why you'll find that organism has a high affinity for the lungs. Okay, we also have pseudomonas. Then we have microaerophilic. This bacteria that can grow under very low amount of oxygen. Okay, so and you'll find microaerophilic organisms sometimes are also found in a category of um, obligate anaerobes because they normally don't, they can survive without the oxygen. Okay, so this is what we're saying. Bacteria can have aerobic or anaerobic and obligate macrophilic, obligate anaerobe, facultative anaerobe. Okay, so in terms of pathogenesis, bacteria is basically bacteria getting to the blood, causing bacteremia that will be systemic. However, we have some also local infections by bacteria, for example, that can cause a boil or other things. So for the bacteria to grow well, they require nutrients, oxygen or absence of it, depending on which type, a neutral pH, and an optimum temperature. Okay, so